Good morning to all. It seems like scholars like us, while in the journey to reach our goal, there can be a long way to the end. My strong belief is an event like Bioinformatica Indica can shorten the distance. So dear friends, the Bioinformatica Indica is the platform for us to get knowledge, to improve our research career, also to give innovative ideas and right focus towards future. I welcome all of you for today's session. To give valuable remark for today's session, we have Dr. Vinod Scariya with us. I request to Dr. Umman V. Umman to chair this session. Dr. Umman V. Umman is an emeritus scientist of CSIR Government of India. Now he is the chairman of Kerala State Biodiversity Board. He had around 35 years of teaching and research experience. Professor Umman's research contributions are in the field of biodiversity, taxonomy, comparative endocrinology, development biology, and animal physiology. He is the author of more than 135 research publications and several reviews and chapters in peer-reviewed national and international journals, which earned him the fellowship of National Academy of Science and Kerala Academy of Science. Professor Umman is currently Honorary Scientist of Bioinformatics Center of this department. I welcome you, sir, to chair this session.
My colleague, Dr. Chuchanger, Dr. Pavanthar, other distinguished scientists from various parts of our country, and my dear students. Good morning to every one of you. As I stand before you in this beautiful auditorium, which is inaugurated for Bioinformatica Indica 2014, I feel so proud and happiness in my mind that the vision of a small Center for Bioinformatics of Dr. Achyut Shankar and I has been materialized to this level in early 2014. Probably Dr. Achyut Shankar and I can only share that thought, that how we started, how we progressed, and where we are today. The present generation students in this building will not even realize the importance or the glamour they have today. They think it's all, take it for granted. Only one person, and I also realize that, Dr. Achyut Shankar can only imagine the path, the road which we came through. It was not all this glamour with a very beautiful hall where you have the privilege to address and the privilege to listen to in early 2014. The road was very rough, but thanks to everybody, even the funding authorities, God Almighty, the blessing showered upon Dr. Achyut Shankar in translating his thoughts into a reality in the last 8 years' time. Several scientists joined in between the bandwagon. Pavan also was here for some time. He has gone for better pastures, and he is now well placed in Delhi, which is certainly good for him. But he still comes back and maintains his relationship with this with this department. Bioinformatica Indica 2014. I was just verifying with Dr. Achyut Shankar that come January even years, it is Bioinformatica Indica for this department. That's very good. Because the fact that I could not attend the inaugural function was, I went to attend another conference in Trichy, where that was a 32nd annual meet. It's an annual meet. Here it is once in 2 years. 32nd annual conference of the Society for Reproductive Biology and Comparative Endocrinology. I joined the society in 1983. Since then, out of the 30 conferences, I only missed 2 conferences. when I was not in the country. Otherwise, I attended all the 30 conferences in my association with the society for 32 years. So which means Bioinformatica Indica will be a forum for the scientists of bioinformatics and computational biology to remember every January even years, so that's the time to prepare yourself for Bioinformatica Indica and come to Trivandrum to share, exchange notes, to share new information which you gathered in the previous 2 years. So I'm very happy that Bioinformatica Indica took its place in the history of this department. I'm very happy this morning to chair this session, though I'm not the apt person to chair a session on genomes and genomics. But the very fact that the marriage between biological science and information technology, it's going to revolutionize the research in biological science and particularly in translational research. The fact that you are going to listen to a young, dynamic, handsome scientist from CSIR Institute of Genomics and Integrative Biology is basically a graduate in medical science turned a scientist, which is usually not very, very common in our country. In the West, particularly in the US, most of the scientists are MD, PhDs. And that kind of a trend is now happening in our country too. An example is Vinod Scariya. So the translational research, the bench from bench to bedside and back, it's a bidirectional road. So the experiences on the bench is translated on the bedside, and information gained on the bedside, it comes back to the bench to modify or to discover something new. So the drug discovery is the norm, or is the talk of the everywhere you go for a integrative biology conference, you listen about drug discovery, because we have a vast biological diversity in our country, particularly in the Western Ghats and also in the Eastern Ghats. Western Ghats is a treasure of several thousands of plants, and many of them probably have got answers some of your ailments in the nature. So nature gives you ailments and nature shows you the treatment also. The fact that this department published a paper last month on a traditional knowledge translated into it could be translated into a product by the pharmaceutical companies if they wish to, or the anti-venom effect of a plant used by tribals for snake bite victims. So we have a treasure of plants to be protected in the diversity of our country in the Western Ghats. And I am given the responsibility temporarily to look after the biological diversity of the state, and we are in the process of looking after the biological diversity. I don't want to take more time. This morning I have immense pleasure in formally welcoming and introducing the speaker, Dr. Vinod Scariya. He is a scientist at the CSIR Institute of Genomics and Integrative Biology. He is a clinician turned computational biologist. That's what I talked about. He's a clinician turned computational biology. Welcome you to the world of students of science. With research interests in translational genomics and genome informatics. He is interested in understanding the function and organization of non-coding RNAs. We never heard of it when we were students. It's a new information for us. And how genomic variations could potentially impact them. There is nothing not needed in nature, actually. Non-coding, we did not even know about non-coding RNAs. If at all we knew that they are not functional, if they were not, but that is because of our lack of information. So as we develop modern tools, we begin to understand more functions. And nothing is not without any function. He is also involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of genomic variations. He has adopted novel and creative strategies like the use of social media and participation of large number of undergraduate students in collaborative projects to accelerate genome annotation and co-creation resources for genome annotation. In fact, the State Biological Diversity Board, with the help and technical support of Achyut Shankar S. Nair and his brother, Dr. Uday Shankar S. Nair, who is a professor in Alabama University in Huntsville, US, is embarked into a program of mapping the biological diversity of the state through smartphones, crowdsourcing. I do not know much about it, but then Dr. Achyut Shankar and his brother are helping us with that kind of a crowdsourcing, with using smartphones, looking at or understanding the biological diversity of the state, which is probably very easy and correct rather than you map it from a remote sensing satellite, which has created some kind of confusion in the state of Kerala these days. And Dr. Vinod Scariya has more than 75 research publications. I congratulate you for that. You are a young researcher, so he has several publications to his credit even at this young age. I must congratulate you for 75 publications. Usually you attain only when you become a professor or even after that. So I am very happy to invite Dr. Vinod Scariya to talk on personal genomes to precision medicine. Over to Vinod. I am very happy that I am invited this morning to chair this session, thanks to the organizers, particularly to my colleague and close friend Achyut Shankar.
A very good morning to all of you, and thanks Professor Umman for the nice introduction, and Professor Achyut Shankar and the entire team for inviting me to present here. Well, before I start my talk, I would probably introduce my lab. I work in the general area of genome informatics, essentially anything DNA and RNA. The three main areas that I work on, the first is to look at the non-coding RNA biology, which is not what I'm going to present today. The rest of the lab actually works in two interesting aspects. One is to understand how the vertebrate genome is organized and how they are regulated, and also try to integrate both of the information into looking at clinical applications. Well, the basic premise we are here at the moment, and which makes it very interesting, is the cost of genome sequencing, which has drastically come down over the last 1 decade. Right? When the international consortium started looking at the human genome, that was around $3 billion to do a human genome. And right here in 2014, this is only 2010, in 2014, we stand at approximately around $2,000 for a human genome. I mean, that's a huge and drastic improvement in the throughput, and of course the drastic reduction in the cost. Now this opens up a lot of challenges. The first challenge is that, of course, you would have a large number of genomes, but that's also an opportunity because you could use these genomes for clinical applications. Right? And there, of course, there are a lot of people who have done the human genomes and made it available in public resources, like these people, including Mentor, Watson, Mishan, Christ, and a number of people as part of the personal genome project. Well, the problem starts here. Most of these genomes that are available in public domain are actually from white homogeneous populations. So you can take about the Mentor and the Watson populations of Caucasian in origin. You could take the early Asian genomes, which are the Han Chinese and the Korean, which are also quite homogeneous populations. You could also have the African populations, which are sequenced, also from quite homogeneous populations. Well, what you see here is actually the entire diversity of the world populations in terms of genomic variability. And as you see here, the most diverse regions of the world are in Middle East and the Central and Southeast Asia, and of course the Americas. Well, as you see here, there's a huge gap in the available genome information in this part of the world. Well, that's when we started in 2009, and we had to learn how to do genome sequencing and of course do the analysis, and that's what we did. So in 2009 we started with a collaborator Sridhar in my lab, a close colleague of mine, and one of the students in Sridhar's lab, Ashok. We embarked on trying to actually understand from scratch how to do a genome sequence and go about doing the functional annotations in terms of trait associations, in terms of annotating the protein coding as well as the non-coding variations. We ended up publishing this paper later, and from there on we actually tried understanding the entire diversity of this region, which was a gap in the understanding of genomic variations. We tried with the Sri Lankan genomes, neighboring countries, the Malaysian genome, the Philippines genome, and now we have practical collaborations from the entire part of this world. Well, now you can do human genome sequences, but do we really understand how to go back looking at it? So that's a major challenge. So if you look at the human genome, which is assume that this is a pie of around 3.3 billion bases, what is functionally annotated in terms of that could be understood in terms of variations and the functional effects is practically limited to 2% of the genome, which is essentially protein coding exons. While today what we know is a large proportion of the genome, a number as variable as 40% to 60% actually transcribes for RNA. They don't encode for a functional protein, but they do transcribe for RNA and what is presently known as non-coding RNA. A very small percentage of this entire non-coding RNA set, approximately another 2% of the genome, we now know encompasses two large classes depending upon the size. One is a smaller non-coding RNA class, which includes microRNAs, no-RNAs, and so on, which most of you know, and a recently discovered class of longer non-coding RNAs called long non-coding RNAs, which also encompass a lot of non-coding RNAs which have previously been characterized as antisense, process heterogenes, and so on. Now, this large class of long non-coding RNAs have expanded in the recent years, mostly in the last 3 to 4 years, and today encompass the largest gene family known in the human genome. Right? This approximately encompasses 28,000 genes, around 56,000 transcripts, quite larger than what we knew about the protein coding genome. Well, now the real challenge is how can we actually look at variations in this non-coding RNA component, at least under 2% of a non-coding RNA component, which we know some annotations exist. So we were one of the earlier groups to actually look at variations in this non-coding RNA. We started with microRNAs as early as 2009. We have a series of publications where we looked at variations which can affect microRNA biogenesis, variations which can affect the seed sequence, or variations which can affect the target sites. We also started working on other classes of smaller RNAs, including snow-RNAs, which are involved in RNA modifications, which have a peculiar structure and a peculiar conserved motif. So you could have variations which could disrupt the structure or the motif. And then we landed up with a newer class, which is long non-coding RNA. Well, when we started with this long non-coding RNA class, practically nothing was known about the functions. And even now we don't really know about the functions of the majority of this long non-coding RNA. So we started understanding whether we could look at long non-coding RNA structure, we could look at long non-coding RNA processing, and of course long non-coding RNA protein interactions. We have a series of papers describing how we could do that using experimental techniques. I'm not going to delve deep into it. Well, what was interesting and what gave us the confidence that long non-coding RNAs could be functional was to actually look at unbiased genome-wide data, which is available in public domain. A lot of people have done genome-wide association markers for multiple traits and diseases. We asked this question, now if you take the genome-wide association SNPs with a quite good statistical significance, where do these fall? To our surprise, what we found was a large number of them, as expected, do fall in protein coding genes, but to our surprise what we found was a quite significant number of them fell in long non-coding RNA regions, and some of them were ambiguous in the sense that they could also fall in long non-coding RNA regions and protein coding regions as in the sense of antisense. So we asked this question, the general balance in the community is that long non-coding RNAs are not functional. Now, the argument people use for the lack of functionality is that there's a large number of variations in long non-coding RNA, or in other words, long non-coding RNAs are not conserved. Right? So we actually went back and looked at this information. We could really find that these are the long non-coding RNAs, and this is the density of variations in long non-coding RNAs, and these are the protein coding exons, which has a very small number of variations compared to long non-coding RNA classes out here. But now if you actually look at functional sites in long non-coding RNAs, so some functional sites that belongs to exons in a gene, in a protein coding gene. You could have introns and exons. So the exons are the functional sort of motifs or functional parts. So you can actually functionally annotate long non-coding RNA regions, including sites which do process into smaller RNAs, sites which do interact with proteins, and sites which would interact with other RNA classes or other DNA classes. So when we ask this question, can we actually look at variation frequencies in this functional elements and not just entire length of the long non-coding RNA? And to our surprise, what we found was that the variation density of the functional regions become almost similar to that of protein coding exons, or in other words, the conservation is limited to very small functional elements in the long non-coding RNA, but not the entire length of the long non-coding RNA, which we recently published. Now, so now what we have is we have a handle towards looking at long non-coding RNA variations and actually prioritizing the functional variations which you could experimentally validate at a later point of time. Now, assume that we understand the variations and their functional impact. The real test of the game is going to happen in here. That's the clinics. Well, I mean, as trained as a clinician, most of my life has gone into looking at the right diagnosis by eliciting the right questions, eliciting symptoms and signs, and of course ordering for diagnostics to arrive at the right diagnosis. Right. Well, most of the treatment actually happens on an average patient. I mean, this person is not an average person; this is a human being, right? And each of them are individual. But nevertheless, the entire modern medicine actually works on the concept of an average patient, which is for why you have clinical trials. Which is quite paradoxical because the founding father of modern medicine, Hippocrates, very early had stated it's far more important to know what person the disease has rather than what disease the person has. It's just an oxymoron because what essentially modern medicine does is complete reverse of what he had said. Now the question is, can genomics be applied to actually help people and to actually give the right diagnosis and the right treatment? Now the problem with the reverse of this, which is happening in modern medicine, is that once upon a while you get treated for an average patient and patients develop things like this. I mean, as bizarre as this, these are nothing but adverse drug reactions because you treat the average patient. Now, adverse drug reactions is not as rare as we all believe. Even in a country like India, around 3 to 4% of people who land up in an emergency medicine department end up with an adverse drug reaction, which is also a large cost in terms of the number of patients. So we went back and asked this question, is there any evidence or is there evidence where we can pull in from genomics to avoid these complications or adverse drug reactions? And this is actually a fledgling new field called pharmacogenomics. Pharmacogenomics is nothing but genetic markers which can influence the predisposition, the metabolism, excretion or the action of drugs. Right? Now we actually went back to literature and asked this question, are there enough literature to actually do this in clinical practice? And what we found that yes, we have a large number of papers, almost growing at the rate of 2,000, 2,500 papers in the last couple of years. Now the real question is, yes, if you have papers in this field, can we put it back into a usable, searchable, interpretable format which can be used in clinical practice? So we went back and looked at other databases which has collected this. Of course, there are a large number of databases which collected information, but what we figured out was most of them are not comprehensive enough or not comparable, encompassing all the data that's out there. So that's when we started thinking we should actually do this all by our own, but of course you have limitations in resources. So we actually employed a crowdsourcing approach. So we created something called the OpenPGX Consortium, which includes researchers from mostly Southeast Asian region, including a lot of our collaborators who did personal genomes, to be able to create a compendium of evidence, clinical evidence, published in literature, to be able to interpret personal genomes, to be able to avoid such complications. So we trained around 100 researchers over the last 2.5 to 3 years, and what we have is at the moment a large compendium of resources which includes one of the most comprehensive maps of transport and metabolism, around 25,000 interactions, and around 6,000 annotations for pharmacogenomics, which is roughly around 30% of the entire compendium of data that's available out there. Now each of these datasets actually go to multiple different QC processes which right from the annotator, which is mostly a student or a graduate student, right to the subject expert who is a clinician, who is an expert in the field. Now, after you collate the data, what you end up as is actually quite complicated, right? So, I'm sure nobody could make any sense out of this. There's nothing but all the drugs out there, all the targets out there, and before the drug reaches the target, you should either have it transported into the cell or get metabolized. Now, this is quite a complicated network which most clinicians would not want to see, right? This is not what a clinician would want to see and interpret when you look at a personal genome. So that's when we decided that we should actually develop tools and resources to be able to interpret this complexity of data based on a personal genome. And that's when we collaborated with one of the groups out there in Malaysia, Professor Zaki. We've also collaborated with the Malaysian genome. So the idea was to actually implement a computational framework where you could actually use the complexity of information, make it simple, analyze it and make it simple in a visual format that most clinicians would understand. And what we ended up is something like this. Now this is quite simple for a doctor to understand because doctors understand drugs by the classes of drugs, cardiovascular drugs, anti-inflammatory drugs, anti-infective drugs and so on. Doctors know a bit, not that bit, but quite a bit about drug transporters, metabolism and targets of action. You could actually color them. So if you have a colored gene here, that means that you have a problem out there, and if you have a non-colored gene which is gray, that means that you don't have a problem out there. So you can actually follow each of this drug and see, okay, if it lands up here, you have no problem. If it lands up here, you have no problem. Then it will go to the target right there. So it's fantastic. You could have a class of drugs which could be metabolized by this particular, transported by this particular gene. And yes, so you have a problem here for this subset of genes. So you should not prescribe this subset of genes for this particular patient, right? Which makes life much more simple. The other application of such a complex data resource is to actually use population scale data sets, which was generated for something else, to be able to understand population scale differences in drug predisposition or rather pharmacogenomic effects. So in this particular problem, what we did is we took all the pathways for clopidogrel metabolism. I landed up in the lab of one of our collaborators, Dr. Dwaipan, who runs one of the largest diabetes consortium projects in Asia, with a large number of samples which were typed as part of the Indica consortium for diabetes. Now these are cross-sectional samples of a large population of North India from multiple different states. You could actually go back to the database or the pathway, pull out all the genes and the variants, and be able to look at allele frequencies of these variations in this population, right? And what you see here is this is the population frequency, which is the size of the bubble out there, and these are the populations out there. So what we found, which was quite interesting, is that most of the North Indian populations do have quite low allele frequencies for most of the variants which are associated with clopidogrel metabolism, which essentially means that Indians in general, or North Indians in general, would require a lower dose of clopidogrel compared to what most of the Caucasians would have. Now this could translate into a lot of savings because, and of course avoidance of adverse drug reactions, because most of the people who go into a stenting procedure end up getting a higher dose of clopidogrel because that's what Caucasian population needs, while Indian population requires a much smaller dose. So you have the problem of an overdose there. Now this is all fancy out there, but how can people know that you can do human genomes, and how can you benefit from doing human genomes? So that's when we started this website. It's practically a website called meragenome.com, which essentially means my genome. It's an educational program or an educational website which gives you quite some information about what a personal genome is, how can you do a personal genome, what can you interpret from it. Also you can, for health professionals, of course doctors, are the benefits of doing personal genomes and so on. So we decided to run an interesting survey on this website because there were quite a large number of hits coming to this website for people who wanted to know what personal genomes is. So we gave them 4 options. So the first option was you could pay for your full sequencing and analysis and keep it private. The second option was you could do the sequencing for free, but you pay for the analysis, which is a part option. Option 3 is you could You could get a free genome sequence and analyze, provided you reveal your name information and put it in public domain, and of course fourth, where you can opt out of the entire program. Now, the basic belief in the community is that people would not want to do personal genomes and put it out there in public domain, because there are a lot of ethical legal issues to that. And we ran the survey on around 120 people, and what we got was absolute reverse of what we believe. Most of the people, around 87% of the people, would be willing to share non-anonymous data in exchange of sequencing and analysis. And the interesting part of this is most of these people do not have a familial or a genetic disorder, which means that they are quite normal people who are willing to do human genomes and make it available in public domain for free, provided they don't incur a cost. The other interesting thing is most of the people who have a genetic disorder or a familial disorder would not really want to reveal the information out there, which is also not quite surprising because they would have to face ethical and legal implications of that. So that necessarily means that we are in a light situation where you have at least education among the so-called elite people or educated people, the benefits of doing human genomes even for normal people. The other opportunity to look at genomics in clinical applications is actually look at rare disorders. Rare disorders by definition are people who have a disorder which occurs 1 in 50,000 or 1 in a million people. In India we have a unique opportunity there because India contains a large number of endogamous populations with peculiar marriage practices which forbids many of these communities from marrying outside the community. So you have rare diseases which run in families or rather in communities, which offer a new opportunity to look at rare disease genes, right? And the opportunity of looking at this rare disease genes is that you should be able to identify the gene using the genomic sequences that are available in a quite easy fashion, and be able to model in a model system, and of course be able to screen at a later point of time. So the procedure runs where the doctor refers a patient who is an undiagnosed or a quite rare disorder for which the gene is not known. We will be doing the genome sequencing and of course giving back the report saying that you have a novel variant or so. And you have an opportunity where you can actually model this in a model system, for example zebrafish, where one of my colleague and a close friend works. So I'll give you one example of such a case, and this case actually came from the western part of India with a peculiar skin disorder called epidermolysis bullosa simplex. So essentially you have bullae on your skin or the palmar surfaces or in the feet or wherever you have applied pressure, and the bullae break down and get infected and get disfigured, right? And this is an autosomal dominant disorder, quite well known. So these patients came to us because they have tried for almost all the variants that are known for epidermolysis bullosa. They have gone to a US lab, got it done, but they couldn't actually identify the variant associated with known variants associated with this particular disorder. So we went, gave them a visit, and to our surprise what we found was an autosomal dominant disorder which should not occur at large frequencies like this. You have a large family of around 4 generations, close to 13 people affected with this particular disorder, and this should have been prevented long back. So what we did was we did an exome sequencing of 2 individuals. We found a novel variant out there which is in the creatine 5 gene. We went back, sequenced the probands using standard Sanger sequencing and showed that the probands do have the variants and not the other ones in heterozygous condition. So what we have is we have a novel variant out there which we can now do prenatal screening for all the individuals who wish to conceive at a later point of time. And this is very important because it's an autosomal dominant disorder where you have a 3 by 4 chance of getting the disorder in the next offspring. Now of course the next opportunity is whether you can actually take this mutations, model it in a model system and be able to screen. And my colleague Sridhar with his collaborator Steve has been able to set up the system where he has been able to use talents which are essentially nucleus domains. You could modify particular single nucleotide variations in orthologous genes in zebrafish, be able to model the disease, and of course the ambitious plan is to be able to screen for small molecules for this particular disorders. So eventually what we foresee is with personal genomes you would also have personal fishes with you which people would carry with them. Well, coming to the screening part of it, what we have been trying to do over the last few years is actually be able to use publicly available data that's out there, to be able to learn from the publicly available molecules and their activities, to be able to predict new molecules and new molecular activities. So this was initially run as part of the open source drug discovery program, also with help from the Garuda CDAG grid computing resources, involving over 250 participants, mostly students. We trained them about basic cheminformatics skills using machine learning approaches to be able to predict new activities or existing activities on new sets of molecules. We have been able to publish quite a large number of papers in this field in the last 3 to 4 years. Now that's human genomes, but genomics is not really limited to human genomes. You could also have genomics of the microbiome, which is increasingly becoming important, of course, the pathobiome, which is the pathogen organisms which reside in the human body and cause diseases. And that's we ended up with microbiology. Of course, microbiology as we know today was originally invented by this guy over here, Anthony von Leeuwenhoek, who was officially a merchant and a municipal official, and by hobby a scientist. Right? And an incredible device he built was an handheld microscope. Right? And I mean, it's surprising to see that the microscope was initially a handheld device and eventually grew to encompass large rooms, while most of the electronic devices that we think of today were initially large machines and became handhelds at a later point of time. Well, the problem with this field of microbiology is that most of the microbiology is still limited to microscopy or phenotypic markers, with limited methodologies to look at culture without culture and isolation. So most of the microbiology that we know today is essentially limited to the way you can culture it, the way you can isolate it, and the way you can phenotypically screen it. And that's quite a big problem, because in clinical settings, if the doctor really doesn't know what the infection is, you cannot really identify the pathogen in most of the cases, at least in the bacterial cases. And there are very few things that most clinical microbiologists will never tell you. One is the clinical stamp is sterile, and this is a really big problem because, I mean, assume a person who has has a mass in the brain, right? This could be infection, this could be cancer, and the only thing doctor would want to know is this sterile? He doesn't even want what organism is in there, right? A tumor is supposed to be sterile, but an infection is not supposed to be sterile, right? So it's a 0-1 question, but most clinical microbiologists would not be able to answer that. The second question is it only one pathogen or two pathogens or multiple pathogens? And this is very important in ICU settings today, because most of the infections occur because of multiple pathogens of multiple sensitivities. So we used approach of genomics. I think I've run out of time probably. So we used the genomics approach to understand diseases for which we don't have approaches to culture, isolate, probably we don't know what the organism is out there. So one of the epidemics we investigated was this epidemic of encephalitis. I don't know if people in Kerala know about it, but there's a quite rampant epidemic of encephalitis which occurs in a part called Gorakhpur in India, which is in the Indo-Nepal border, which practically kills close to 1,000 kids every year of the last 5 decades. Initially it was thought to be Japanese encephalitis, but now nobody has any clue what this virus is. People have done vaccination with Japanese encephalitis, but nothing really happens out there. So we got 2 samples from the National Institute of Virology. Our collaborators were kind enough to give us the samples. The first sample, they knew what it was. In the second sample, they didn't know what it was. They just had an EM of the organism out there, of the virus out there. So we sort of standardized a pipeline where we could actually take the viral supernatant, do the library preparation, put in sequencing, and do the analysis. And initially we started with very simple analysis like mapping it back to all the bacterial, viral, and the human genome. The first case, we were quite fortunate. We just got one peak out there, which was Japanese encephalitis virus. So we knew it was Japanese encephalitis virus, and we got only Japanese encephalitis virus. That means that you can actually take a mixture of genomes out there, do the sequencing, because almost all the living organisms would have either DNA or RNA as the genetic material. So whatever genetic material is out there, you should be able to figure out from the genome sequences out there. But the second was much difficult question where you really didn't know what the organism was. In bacteria, of course, you could do multiple different things like looking at 16S RNA, but what about viruses? There's no way to look at them. That's when we developed this computational approach, which is quite simple or intuitive. So the idea is very simple. You actually take the genome break them up. So this is a simulation out there. So you take a genome, break it up into smaller pieces. You take another genome, break it up into smaller pieces, mix these two genomes in different proportions, plot the coverage out there for each of the schemas, and be able to figure out whether you could distinguish the genomes or not. The surprising fact is at a difference of 1:3 or 1:4 coverage difference or concentration difference or copy number difference, you should be able to isolate multiple different organisms. The other important thing is that anything which falls under this peak or the area under the curve of this peak is essentially going to be the genome size of that organism. So you will have.
What is K there?
So K can be K is a small fragment. This could be anything from 10 mer to 50 mer.
On graph it has to be a specific number, isn't it?
Right.
So the one you have shown here is
It's around 36 mer. So you could have area under the curve and actually just looking at the curve, you should be able to figure out whether it's a virus out there, whether it's a bacteria out there, or whether it's a large genome out there. So each peak out there essentially comprises of one single organism. So even without knowing the genome of the organism, even without even knowing that the genome exists, you could actually do the genome sequence of a mixture, be able to look at peaks and identify the number of organisms out there, and even figure out whether it belongs to the virus class, it belongs to the bacterial class, it belongs to an eukaryotic class. So this is how the graph looked like when we mapped it back to the reference genomes. Unlike the previous case, what you see here is a large number of genomes which map to this particular set of reads. So there are two possibilities here. One is that there are multiple different organisms out there, and the second possibility is that you have a new virus which has homology with each of these strains of viruses. So we used the K-mer approach, and what we figured was it was a completely new virus which had quite large homology with the existing endoviruses, which we called IJB 2009-2. We are quite curious at this point of time whether we can apply to multiple different things out there. So we ended up looking at things like shrimp, looking at plant pathogens, and so on. Shrimp is quite important because India, Sri Lanka, Singapore, and many of the Southeast Asian countries actually grow shrimp on a very large scale and export them to Japan and United States. And one of the diseases which actually wiped out the entire shrimp industry is this virus disease which produces white colored spots out here. It's produced by a set of related viruses called white spot syndrome viruses. So we used this approach to actually take a shrimp early in infection, grind them up, sequence them, and actually figure out what it is. So this is how the graph looks like. So what you see here is a peak out there, it's broken here, so this is shrimp probably because it's a very large peak. And you have a small peak here which looks like a virus. So this should be, you should be able to take this reads out. So we actually took this reads out, so you could identify the reads and take the reads out containing this particular K-mer. And what we found is we could really assemble a almost complete genome of the virus, which is a quite large virus, and be able to annotate them as WSSV or white spot syndrome virus. Yeah.
Shouldn't you end up getting the mitochondrial genome also as an read?
Yes.
Whenever you do it,
Yes.
you do get
Yes.
it.
You do get. And mitochondrial peaks do occur very late in the plot. I don't have the entire plot because mitochondria
I'm sure I entirely follow what is late there. So very high coverage.
very high
low
coverage,
frequency.
very high coverage and low frequency.
And why is that high for?
Like high coverage because for every nuclear genome copy, there are few hundred mitochondrial genome copies. Okay. The next thing that we tried doing this technique was for looking at tuberculosis. Tuberculosis because it's quite prevalent in the community, and the right diagnosis and drug resistance typing actually takes anything from 28 days to 45 days, which is quite long in clinical scale. So most of the people end up in an empirical treatment which actually increases the drug resistance. So we did this application for tuberculosis, and behind that what we have is one of the largest databases for mycobacterium variations at the moment, encompassing 21,000 variations. Many of them are associated with drug resistance patterns, which we recently published at this point of time. So I think I'll stop there. But the real challenge at the moment in genomics is that there is enough data, machines can throw out enough data, but there are not enough people to look at the data. Way back in 2008, we tried looking at crowdsourcing as an approach to bridge this gap by involving large number of students. And this for tuberculosis, which is published in 2009. We did it for Bioinformatics with around 250 students. We published a large number of papers there. This has been part of the open source drug discovery program, and there have been a lot of people who have written about these programs and how crowdsourcing could actually be a cheap and efficient alternative to do that. So the idea was to actually systematize this into a platform where people could crowdsource whatever programs they have. So if you do a genome, you don't have the skills to do the analysis, or you did the initial analysis but you need a large number of analysis to be done, you could actually crowdsource it into a portal what we call as crowdomics.org. We ran this last year with around 15 programs. We are relaunching this again this year with around 20-25 programs. I'm sure a lot of people in the group would recognize their faces out here. I think I'll stop there, but before somebody told me if you don't understand anything, probably try doing your best. Well, before I take the question, most of what I talked here is actually work of a lot of students and collaborators out here who have helped me do the experiments. I'm not an experimental biologist at all.
So if there are any questions, Vinod should be happy to take them.
Can I just ask?
Yeah.
So when you're talking about that graph, my voice louder. You know that very nice graph you showed clinicians.
Right.
You know that very baby
Right.
graph
Right.
that you have deconvoluted into
Right.
something I understand. At that bottom level you had variations.
No, that is the map for one individual.
Right.
Right. The bottom panel is actually the drugs and the drug classes.
At some stage you talked about variations. Yeah, that's
Yeah.
right.
This is the drug classes.
So when you look at variations here,
Right. I don't see variations here. I see only whether the gene got disrupted or the gene doesn't get disrupted.
okay, so my question is that how do you differentiate between, when you do your sequencing, you have these silent SNPs and you have the SNPs that change into the amino acid itself, right?
Right.
They're the silent ones.
Silent ones.
Both have effects, don't they?
Right. Right. So at the moment we don't really look at the silent ones. We are looking at only ones which have evidence. Right. Right. So we are looking at ones which are non-synonymous and deleterious. We are looking ones which are regulatory, and what we found was most of the genome-wide association SNPs for the pharmacogenomic markers, by doing whole genome sequencing, will actually figure out what was the causative variant, either regulatory or causing the protein change. So what the doctor would want to know is not the RSID of it. They would just want to know whether the drug will go through that pathway or don't go through that pathway.
And all this information is from annotations.
All this information, the background information is from the annotation, right? It's from the complicated map that you saw here.
Yes.
It's actually a subset of this map, but now browsable, so that you could actually mouse over this particular drug and see what this drug is. Still complicated.
But it is a dynamic map, right?
Yeah, so for every individual you can create this map.
And then annotation does keep changing.
Yeah, so if the annotation changes in the back end, this gets updated in the front end. That's right.
You showed a graph in which 36 MERS were being plotted, frequency of 36 MERS. What is your insight about the occurrence frequency of this big MERS?
Right.
Did you notice that at any particular range of the size of the MERS that they show up some hidden characteristic of the viral genomes?
No, so the K-MER is essentially a way of computationally deconstructing the genome. I mean, I'm not taking it in the assembly parlance of it, right? Here I'm taking as a way of deconstructing the genome, right? So I need a K-MER which is large enough so that they don't occur in quite large number of repeats. Right? I need a K-MER which is small enough so that I have enough occurrences in a read, right? So if I have a read which is 100 base pair, I could get comfortable with a K-MER of 36 base pair. But if I take now a K-MER of 100 base pair, every read is a K-MER, right? So I will not find a duplicate of that K-MER in another read unless I have very large coverage, right? So it's a trade-off between coverage, the trade-off between the kinds of organisms you're looking at, and so it's actually by practice for a read length you sort of devise for a complexity. This is the K-MER that is most likely.
Gives ADMET profiles. ADMET.
This is actually ADMET profiles decided by the genomics.
Okay, different people have different metabolic profiles.
Absolutely. So what you
So
see,
that's your next slide.
absolutely. What you see here is essentially that.
So this include that one.
Absolutely. So what you see here is that admit,
Okay, thank you.
but admit for the population.
This is a general question. As of the present status, how long it will take a common man to carry his sequence while going for a checkup in India?
So if you are ready to pay for it, it's already there. If you can pay for it, it's already there.
It's very expensive.
Right now it's okay.
So at the moment, the whole genome sequence would cost around in Indian rupees around 2 lakh rupees, and the part genome or the exome would cost anything around 60,000 rupees to 1 lakh rupees.
So Anisha can afford. Anisha can afford.
5 years, I think.
And another year, you should think the cost of it to be half. I mean, every year it becomes almost half. So another 3-4 years, you'd be
Oh, then
quite
you should
affordable.
wait for a few more years to get diseased.
Yeah, but the problem with that is you could always wait till it becomes zero or tending to zero.
Yes, please.
Doctor.
Yes, please.
So when you talked about those viruses, and you have an association with the disease that you're looking for, right? Like that Gorakhpur disease. So as a doctor, with recent evidence about the importance of the relationship between our bacterial constitution and the rest of our processes like inflammation and all, that there is a certain minimum concentration required
Outside the body.
bacteria.
Outside the body. Outside the body.
No, I was asking you, so does that hold true for viruses as well? That do you want to eradicate everything in the body, or is there a level above which it starts becoming a nuisance and you just keep it below that?
Okay, so it's a very interesting question. So the way most of the higher organisms are built, including humans, the organ systems are supposed to be sterile. So if you take blood today of a normal person, I'm not normal, I have throat infection, but I mean apparently normal person, and if I do this, I will not find anything other than human. I should not find anything other than human. Right? Now, if I find anything other than human, that means that there's a breach in the immune system. That means you have an infection. Right? Because if the concentration is quite low, the body would eventually take care of it. But if you find it in a recognizable quantity, that means that there's a breach of the recognition system somewhere, that you have enough concentration. That's essentially infection. Right? So if you take any body fluids, whether you take a pleural fluid or urine for that example, it's supposed to be sterile.
Anybody has the last question? Yeah, this one has.
In one of the slides in the beginning, you mentioned about the microbiome.
Right.
So, as a non-life science person, what is most shocking to me is that you have more number of cells in the human body than the human cells itself from the microbiome.
Right.
So, is it that at the center of the whole story that you place the human genome and you talk about variations in it and mutations in it? Should we actually replace it with the microbiome and then rework the picture?
Okay, so the real answer to this is not everything is controlled by the genome, right? But there is a significant amount of things that are controlled by the genome of your system. I mean, when I talk about the system, I talk about everything covered by the skin, right? I mean, assuming that a human being is actually a tube, right? And anything in the wall of the tube is actually the human system. Even your gut is, actually outside the system. Right? It's a tube running from bottom to down there, right? So there are things which the system does, which is influenced by the system genome, and the things that are outside, which includes the skin, the gut, which also influences what happens inside, right? So both of them are important. You cannot solve all of them by just looking at the human genome. Absolutely right. But there's a significant amount of things that happen in the system, like for example drug predisposition, which is decided by the human genome. Nothing else decides that. But when you talk about absorption, yes, since it has to go through the gut, you need microbes there. But if it's an injectable out there, you don't really care whether what is the organism there in the gut.
Do you think the meragenome.com will revolutionize the medical practice of our country?
No, it's just an educational website. We just want a lot of people to know what is the hope of genomics.
So thank you, Dr. Vinod Scariya, for opening the doors of revolution in medical practice and probably drug discovery in the years to come. Thank you very much.
I'm sure that everybody enjoyed the talk and it was very informative talk. Thank you, sir. Now I request to Dr. Uman to give the token of her gratitude to Dr. Vinod Scariya.